And how are you? I'll just say hello to Elise. Hello, Elise.
Hello, how's everyone?
Good, thank you.
That's good.
We ought to say hello to Kerry, who's joined us already. Hello, Kerry. Good to see you. Thank you for doing the presentation. Rob is with us, but who's going to be chairing the meeting, but we can see Rob now, so good.
Hello all. I am going to start the ball rolling. I will just start, if I may. I know there might be some people who are not familiar with the Zoom platform. So I'll just explain a little bit how this works. If you take a look at the main screen, down at the very bottom left-hand side of the screen, you can see that there is an option to mute your microphone. When we are having a group webinar like this, it's a great idea, please, if everybody could mute their microphone. And later on, we will have some opportunity to ask questions from our speaker today. And later, when we do that, there are two options to unmute the microphone. You can either click on that icon again, or if you're sitting in front of a keyboard, you can simply press the space bar, which is going to act similarly to pressing the button on a walkie-talkie, which will temporarily take your microphone off mute. We do this just to really respect the other listeners online and our speaker, so as to avoid any background noise. Before starting proceedings properly, I know that we are spread both all over the country today, and I believe we have an attendee, certainly with a Californian address and email address. I don't know if they are in Australia or joining us all the way from the US, but welcome everybody to this first virtual webinar hosted by the Food, Nutrition and Analytical Chemistry group here in Victoria of the Royal Australian Chemical Institute. Since we are spread all over the country, I'd like to make I suppose a more generic than usual acknowledgement of the traditional owners from the lands that we are all joining and meeting today. And of course, we wish to pay our respects to their elders, past, present and emerging. Now, I would like to introduce our speaker today, who has very generously offered her time to give us what I understand or what I believe is going to be a really interesting webinar today. So it gives me great pleasure to introduce Kerry Wilkinson, who is a professor of oenology in the School of Agriculture, Food and Wine at the University of Adelaide. This sounds like a marvelous role, Kerry. I'm slightly jealous because this, as you describe, is a role which enables you to combine your passion for both wine education and wine research. Kerry teaches into the Bachelor of Viticulture and Oenology, as well as into the Master of Wine Business. And she has led the development of an award-winning online Wine 101X course, which sounds just wonderful. Kerry leads a productive wine science research group, and her primary interests concern flavour chemistry of grapes and wine, which we're going to hear something about today. And this spans from management of green characters in the vineyard through to improved methods of oak maturation in the winery and consumer preferences for different styles of sparkling wine. But Kerry is best known for her research into the impact of bushfire smoke on grapes and wine. And this is quite an impressive statistic. Kerry has published approximately half of the entire world's literature on smoke taint to date. So it gives me, like I said, great pleasure to introduce Kerry to us all, welcome you to our our group for today, and I'll now hand over to you, and we will speak to you very shortly. Thanks,
Thank
Kerry.
you. Just share my screen so everyone can see my slides. Beautiful. So I'll start with a little bit of a story. About this time 15 years ago, I was a postdoc at Curtin University based in Margaret River. And it was pretty well about this time of year that my boss and I were invited to travel about an hour inland from Margaret River to the Pemberton wine region. And we visited a winery who wanted some advice on one of their wines. And when we arrived, we were shown through to their winery, and the winemaker poured some wines into glasses, and we stood around for a period of time tasting this wine. And the winemaker explained that leading up to vintage that year, the state government had undertaken a number of prescribed burns to the south of the Pemberton wine region, and that during those burns, the way that the wind works in that part of the world, it had blown the smoke up through the valleys, and then an inversion layer had occurred in some of the valleys in Pemberton, including where this winery had some vineyards. And so that smoke sat in the valley for a period of time, and they were concerned that they were starting to see some smoky characters that were not so pleasant in some of their wines. They wanted our advice on what had happened. And so when I first started tasting this wine, I sort of thought, not really knowing what I was looking for, thinking it's not so bad. I'm not really sure, what I'm looking for. And the winemaker said, no, just keep tasting it, spitting it out, tasting it, spitting it out. And over a period of about 15 minutes of tasting this wine, there was this increasing sort of ashy character on the back palate that emerged. And it got to the point where it really was quite drying and quite unpleasant. And what I didn't know at the time was, this was the first time, and certainly not the last time, that I was tasting the smoke-tainted wine. And it was the start of what's really become, I guess, a research topic that has defined my career since. And so it's a pleasure for me today to share some of the work that I've been involved in over the last 15 years. So when I first started working on smoke taint, we really didn't have any literature to base our work on. There was one preliminary trial that had been published by the Australian Wine Research Institute a year or two earlier, following a fire on the east coast of Australia. But there really wasn't that base of literature that normally you'd be able to go and have a look at what's been done before. So we were sort of at a real starting point. So we thought the best thing to do first was to basically demonstrate that if you take grapes and expose them to smoke, it actually taints the grapes and wine. So we did this by getting hold of some Vineyard grapes, and we really only chose Vineyard because it was the first grape variety that was available to us in that particular season. And we exposed the grapes to smoke post-harvest for 1 hour. And so you'll see in a picture here, these are racks that we laid the grapes out on, and we were using facilities that were available to us through the Perth Botanic Gardens. And we then made wine from both the control and the smoke-affected grapes. And we looked to apply some chemical and sensory analysis to the resulting wines. Now, not really knowing what compounds we should be looking for, I made use of my background in oak flavour chemistry. So my PhD was looking at some of the flavour compounds that are extracted out of oak wood during barrel maturation. And so we look at compounds like guaiacol and 4-methyl guaiacol, so volatile phenols, which are associated with some of those more pleasant smoky characters in oak-aged wines. And it seemed reasonable that we should measure those compounds as a starting point. So we actually applied an oak analysis to these wines. And you can see in our control wines, none of the compounds that we measured, including guaiacol and 4-methyl guaiacol, were detected. But we detected significantly higher, so it's a bit remiss of me, apologies for not putting the units, but this is micrograms per litre. So really quite high levels of these compounds in our wines made from our smoke-affected grapes. The sensory analysis we did for these wines was just a simple difference test. So it was a sensory panel able to tell the difference between these wines, and we did it based on the aroma of these wines only, because the contrast was so stark. And all of our panelists were able to differentiate the wines without any problem. The smoked wines had really quite noticeable smoky, cold ash, meaty, medicinal type flavours. And I actually made the mistake of tasting one of our smoked wines, and it probably took me a good couple of hours to get that taste out of my mouth again afterwards. So this was a really extreme example of smoke exposure of the grapes, and certainly not what happens, in a vineyard situation. So from there, we moved this work out into the vineyard to do some trials. And I should also say, sorry, this work, the very preliminary work that was done by Kristen Kennison, who works for the Department of Agriculture and Food in Western Australia, and this was part of her PhD work. So Kristen then went out and developed this methodology, so these tents that could be constructed around the vines out in the vineyard, and a very, very high-tech metal rubbish bin that we could burn fuel in and then pump the smoke into the tent. And so the first field trial that she did was to expose Merlot grapevines to smoke for 1 hour at 8 different time points during the growing season. So it was around about veraison. So veraison is the time at which the grapes start to shift from a development phase to a ripening phase. So it was about this stage of growth that we had the equipment ready to go. And so she applied smoke at 8 different times from veraison through to harvest. And we also had one treatment where we actually applied the smoke to the grapevines at all 8 times, so 8 repeated smoke exposures. And again, we made wine from those grapes. We had control and smoke-affected fruit, and then applied chemical and sensory analysis to those wines. So I won't show you all of the data from this trial, but this is the glycol concentration that was in those wines. So you can see it's really quite low for the control wine. It was also low at the first time point, so at veraison, and we can see that pretty well every time point after that we saw elevated concentrations of glycol, and we see the similar pattern of accumulation of other volatile phenols that we measured in this study, with a peak at around about 7 days post veraison. So we believe this might be the time point at which grapevines, albeit Merlot in this instance, are most susceptible to the uptake of smoke. But pretty well any time that we had smoke exposure, we were seeing some level of these volatile phenols in the wine. And the chemical data corresponded quite well with the sense as well. So the wine exposure was also the wine that exhibited the most intense perception of smoke taint. What was interesting to us as well is if we considered the concentration of the volatile phenols that we measured in those 8 treatments where there was application of smoke at 1 time point, and we summed them, we got an equivalent concentration of those volatile phenols to what we saw where we applied smoke 8 times. So this essentially demonstrated that repeated or prolonged smoke exposure is also more detrimental than just a one-off incidence of smoke exposure, which perhaps seems a bit intuitive, but it was important for us to demonstrate at the time. Around this time, we were getting some feedback from some winemakers that were suggesting that they had harvested grapes that didn't show any evidence of smoke taint, but then when they fermented the grapes during winemaking, the smoke taint would become apparent. And so based on this anecdotal evidence, with this Merlot trial, we also conducted a study where we monitored the changing concentration of volatile phenols throughout fermentation. And what we saw was this progressive release of volatile phenols throughout fermentation. So initially, just trace levels, really low levels of glycol and 4-methyl glycol in the juice, and then we see this steady increase as fermentation progresses. So at first we thought, well, perhaps this is just the kinetics of extraction of these compounds from the grape skins, but what we saw was a very significant increase after we pressed the skins away from the partially fermented wine. And so between pressing and secondary fermentation, so malolactic fermentation, we saw that the concentration of these volatile phenols continued to increase, which suggested to us that perhaps there was some kind of precursor form of these volatile phenols that was present in the grapes, which might explain why winemakers weren't detecting any smoke taint in the grapes, but were then seeing it occur during fermentation. So this was really the first evidence that there were these precursor forms of volatile phenols present, and we confirmed this via some acid and enzyme hydrolysis experiments. So we took some of that free-run juice and heated it at pH 1 at 100 degrees, or treated it with beta-glucosidase enzymes, and were able to liberate significant quantities of these volatile phenols. So that confirmed there were precursors, and the fact that beta-glucosidase was liberating these volatile phenols suggested to us that at least some of the precursors might be glycosylated. So it was around this time that I moved from Curtin University to the University of Adelaide and established my own wine chemistry group. And so I wanted to continue working on smoke taint and thought, okay, let's pursue this idea of glycosides of volatile phenols. And so what we did was to actually synthesise an authentic standard of the beta-D-glucopyranoside of guaiacol. And this is its mass spectrum. So we can see its parent ion at 285, and there's a solvent adduct at 345. And if we get cleavage of the A-glycone glycol unit and the sugar, we see fragments that correspond to 161 and 123. So what we did was to run this reference compound and we can see it eluted with a retention time of 5 and a bit minutes. And when we ran samples of smoke-affected juice, so juice of grapes that came from, that were affected by bushfire smoke, but also juice that came from one of our experiments where we've applied smoke to grapes, we could see this same peak, the same retention time, and more importantly, the same fragmentation pattern as our reference compound. And so we knew that we'd confirmed that this glycosylation of smoke-derived volatile phenols was taking place in grapes. The Australian Wine Research Institute did some really great work not long after this, where they actually they did an isotope labeling study. So they took a 50-50 mix of guaiacol and deuterated guaiacol and applied this to the leaves or berries of a potted grapevine, and they then went and did some mass spectrometry work and identified a series of glycoconjugates that had that 50-50 deuterated, undeuerated guaiacol unit. So they identified a series of glucose glucosides, pentose glucosides and retinosides that were there. So it wasn't just the guaiacol glucoside that was present, it was actually a suite of volatile phenol glycoconjugates that were being formed. And they now offer this as an analytical method as part of the detection of smoke exposure in grapes and wine. So at the time, after we identified the guaiacol glucoside, we decided to synthesise an isotopically labelled version of this compound for use as an internal standard. And so in collaboration with AWRI, we actually developed a quantitative method based on liquid chromatography, tandem mass spectrometry, which we subsequently applied to a number of different trials to try and understand some of the factors that were affecting the uptake of smoke and the level of smoke taint in grapes and wine. So one of the first things we did was to track the accumulation of these glycoconjugates in grapes. And so we did an experiment where we applied smoke to Viognier grapevines, and we then measured the accumulation of guaiacol glycoconjugates. And we can see that as early as 3 days after smoke exposure, we were starting to see some of these precursor compounds, that concentrations increased to 10, 17 days, and then sort of plateaued after that. So we knew that they were accumulating fairly quickly. One of the questions we were interested in after that was whether different cultivars, so different grape varieties, might be more or less susceptible to the effects of smoke taint. And so we performed what's probably been one of the most logistically challenging trials that we've ever done, involving the application of smoke to 7 different cultivars, so Chardonnay, Sauvignon Blanc, Pinot Gris, Pinot Noir, Shiraz, Cabernet Sauvignon and Merlot. And we did all of this in triplicate, so 7 grape varieties, 3 replicates each with control tents and smoke tents across 2 different vineyards in the one season. So it was a really big trial. And then we threw pretty well every chemical and sensory analysis technique we had at it. And one of my colleagues, who's a plant physiologist, also came out to this trial to have a look at the impact on vine physiology. So that was quite interesting. What we found was that different grape varieties were affected by smoke in different ways. So if we look just at, in terms of, water efficiency and photosynthesis, what we found was that some varieties like Pinot Gris weren't all that affected. So they essentially shut down photosynthesis as soon as they're exposed to smoke. But in the case of Pinot Gris, they're back functioning, as well as the control almost immediately after smoke exposure, whereas other grape varieties took quite some time. In the case of Cabernet Sauvignon, perhaps a week or so until they were photosynthesizing at the same rates as their corresponding control vines. And I think from memory, Merlot took maybe nearly 2 weeks until it was on par with its control vines. So this is my, I apologise now, this is my death by PowerPoint slide, and I've got 2 of them. But this is some of the most important, data that we've generated from our trials. So bear with me and I'll walk you through it. So here we've got the chemical data for some of our grapes and wines corresponding from our varietal trial. So we've got our 7 grape varieties, and then C is for control, so no smoke exposure, and S is for smoke exposure. So in the first column, we've got the concentration of our glycol-glycoconjugates in the grapes, and with the exception of Shiraz, what you can see is in our control grapes, we've got quite low levels in all of the control fruit across those varieties. What that tells us is that Shiraz is a little bit different, and for some reason it has these glycol-glycoconjugates present as natural components. And this was something that we thought we were starting to understand, that this was just this background level of these glycoconjugates, until a trial that we did last year where we had control Shiraz that actually had none of these glycoconjugates. So, now we're wondering if there might be some other, whether it's clonal or regional influences that determine, composition. But we can see that in the case of our smoke-affected grapes, there were significant increases in these glycol-glycoconjugates. And we can see that carries over to the glycol-glycoconjugate levels in the wine as well. So what was interesting is, again, our control wines had really low levels of the glycoconjugates, again, apart from the control Shiraz, but we had elevated concentrations in the wine. So that firstly tells us that not all of this glycoconjugate pool is being hydrolyzed by winemaking yeast or enzymes during fermentation, but it's also really difficult for us to look at the concentrations of glycol-glycoconjugates in grapes and predict what concentrations we might see in wine, and particularly what concentration of the volatile phenols as markers of smoke taint we might see. So if we move to the next 4 columns, this has our volatile phenol concentrations. And so what we can see is, if we just skip ahead to our white varieties, so Chardonnay, Sauvignon Blanc and Pinot Gris, we can see in the Chardonnay and the Sauvignon Blanc and in our Pinot Gris in the controls, there were none of these volatile phenols detected. And at least in the Chardonnay and the Sauvignon Blanc, only really low levels from the smoke-affected wines. So this is in part because when we ferment our grapes, we ferment white grape varieties after we've pressed the skins away. So red grape varieties we ferment in contact with the skins because we want to extract the coloured pigments that are present in those skins. So that partly explains why our smoke application gives us much higher levels of volatile phenols in our smoke-affected red wines. But it was interesting to us because our Pinot Gris, which is a white grape variety, had quite high levels of these volatile phenols. And at the time, the only thing we could use to explain this was the fact that Chardonnay and Sauvignon Blanc were picked at a lower level of sugar ripeness because they were destined for sparkling winemaking, whereas Pinot Gris was allowed to ripen further because it was going to be made into a table wine. Similarly, the Pinot Noir was also destined for a sparkling wine and had lower levels of these volatile phenols than the smoke-affected Pinot Gris. So again, we wondered whether that might have been related to the fruit maturity at harvest. But we can see in all of our smoke-affected red wines, these elevated concentrations of volatile phenols, in particular glycol and cresols and syringol, much higher levels than what we see in the corresponding controls as a consequence of smoke exposure. Again, Shiraz has higher background levels in the control wine as a consequence of these compounds occurring naturally. So if we look at this data, we might initially think that the Shiraz, the smoke-affected Shiraz wine would be the most heavily tainted wine. But if we take into account those background levels, we'd probably actually come back and say it's going to be the Cabernet Sauvignon. And in the case of the white wines, most evidently it's going to be the Pinot Gris wine. And this chemical data actually matches up really nicely with the sensory data that we got. So this is a principal component analysis biplot, which basically takes all of our sensory data and our chemical data and displays it in a two-dimensional format where things that are clustered together have more similar chemical and sensory profiles, and things that are further apart are more different. And what we can see is that the red samples, these are our control wines, and we can see they're predominantly clustered on the left-hand side of the plot. They're being driven mostly by fruit, aromas and flavours, whereas our blue wines, these are our smoke-affected wines, are, at least some of them are being positioned more on the right, and they're being driven by these smoky sensory attributes, so things like smoky, ashy, medicinal, burnt rubber type characteristics, and also the presence of elevated concentrations of volatile phenols and glycoconjugates. So what's interesting is the Sauvignon Blanc and Chardonnay, the smoked wines, sit quite closely to the control wines, and that's consistent with the chemical data that suggested that those wines probably weren't very heavily tainted. Then next we see the Pinot Noir, which was the least tainted of the red wines. We've got our Pinot Gris, our smoke-affected Pinot Gris, which was the most heavily tainted white wine. And then we see our three red wines with the Cabernet Sauvignon, which the chemical data predicted would be most heavily tainted and furthest to the right. So that was quite nice to see that chemical and sensory data match up. One thing that we'd heard for quite some time in the wine industry was that there was this intensification of smoke taint in wine over time with aging or bottle aging. So this is something that we wanted to investigate, and we did this by taking the wines from our varietal trial and cellaring them, and it was probably a little bit of luck, really, if I'm honest, that we kind of put them away after we'd finished our chemical and sensory analysis, and then we remembered that they were there 5 or 6 years later and thought maybe we should repeat the chemical and sensory analysis and have a look, at what's gone on in those past 6 years. So apologies for my second death by PowerPoint slide. The left-hand side basically just shows the chemical data for those smoke-affected wines after we bottled them back in 2010. And we're missing the Merlot wine here only because we used that wine in a subsequent trial, so we didn't have it available to us post-bottle aging. So the data on the right-hand side shows the concentration of the different volatile phenols after that 6 years of cellaring in the bottle. And what we can see is there were small increases in some of the volatile phenol concentrations. So we see from 26 micrograms per litre of glycol in the Shiraz up to 30. slightly higher increase in the syringol. But there wasn't this, really massive release of these compounds, given the concentrations of glycoconjugates that we saw remaining in these wines, after fermentation. And we actually measured the glycoconjugates that were in the wines after bottle aging and found that they weren't statistically different from the concentrations that we'd measured back in 2010. And this is consistent with the fact that at wine pH and ambient temperature, these glycoconjugates are actually quite stable. We really need much lower pH and much higher temperatures to actually, or some enzyme activity to actually cleave and hydrolyse these compounds. So this suggested to me that perhaps these compounds were relatively stable and we weren't seeing the significant increases in volatile phenols with aging. We did repeat the sensory analysis on these wines and that's what was quite interesting. So what we found was, in the case of the lighter styles of wines, so things like Pinot Noir, Sauvignon Blanc and the Pinot Gris, the intensity of smoke taint seemed to increase, but it wasn't so much that it was the chemical compounds that are responsible had increased. It was more that the intensity of fruit character in these wines that's attributed to the grape variety had disappeared over those years, whereas the fruit intensity of the Shiraz, the Cabernet and the Chardonnay had maintained, had remained fairly constant throughout bottle aging, so that the perception of smoke taint in those wines wasn't so apparent. So you could kind of argue that yes, smoke taint as a sensory taint, from a sensory point of view, does intensify with time in some wines, but it's not as a consequence of the breakdown of these glycoconjugates and the release of significant concentrations of volatile phenols. So some of the work that we've been looking at since is trying to understand what are some of the factors that influence the intensity of smoke taint in finished wines, and also what can we do either in the vineyard or in the winery or post-production to try to mitigate the levels of smoke taint. So if you recall from our varietal trial, we found that the Sauvignon Blanc and the Chardonnay that were destined for sparkling wine appeared to have less smoke taint, and we wondered whether that was related to the level of maturity, the fact that they were picked early for sparkling wine production. So we went back and ran another trial the year after our varietal trial to actually have a look at the influence of fruit maturity. And so we did this on 4 different cultivars: Chardonnay, Sauvignon Blanc, Shiraz and Merlot, and we harvested the fruit at 2 different maturity levels. So at total soluble solids or sugar concentrations of 16 to 20 degrees Brix, which corresponds with a sparkling wine, versus 22 to 25 degrees Brix, which corresponds with more of a full-bodied table wine. And what we found with the Chardonnay wine was the smoke-affected wines had fairly similar volatile phenol profiles. So from the chemical data alone, it looked like we would assess these as having equal levels of smoke taint. But when we have a look at the sensory data, what we could see is the dotted line, the dotted blue line, is our Chardonnay wine that corresponds to the early harvest. And so at this time, what we were telling winemakers was, if you suspected your grapes of being affected by smoke, what you needed to do was to harvest the grapes as early as possible, do a quick ferment, and then you could assess the level of smoke taint in that wine. So in this situation with our Chardonnay, if we'd done that, picked the grapes at 16 to 20 Brix, done a quick ferment and looked at the sensory properties, we wouldn't have been able to differentiate them from control grapes, and we would have thought, okay, we don't have any problems with smoke taint. If we'd then gone back and harvested the rest of the crop at the level of maturity to make a table wine and made that wine, what we would have seen is the elevated intensity of smoky aromas and flavours, because for some reason in Chardonnay at least, that's what we saw, that maturity was having an impact. And at the time that we published this paper, there had been a fire up in the Adelaide Hills, and we had an example of a winemaker who came forward and said, this is exactly the situation that we had. We had Chardonnay vineyards. The fire went through, we thought there was some smoke damage, we did our fermentations early, we couldn't see any sensory evidence of smoke taint, so we harvested all of the vineyard block, made wine, and then found we had a smoke tainted Chardonnay. We didn't see the same results in Sauvignon Blanc. We're not quite sure what it was, but we were able to pick the level of smoke in those Sauvignon Blanc wines equally. And whereas we did see some differences in the red wines, because these red wines are fermented in contact with the skins, we're seeing those evident smoke characters in the wines, irrespective of when we harvested the fruit. So one of the other vineyard trials that we've tried to see whether we can mitigate the effect of smoke out in the vineyard has been the application of Kalyn to grapevines. So Kalyn is a clay-based material that sometimes gets applied to grapes in order to protect them from sunburn. So it's essentially a clay-based sunscreen for grapes. So we did this trial where we applied Kalyn to 3 different grape varieties, and we then exposed them to smoke, so with and without that Kalyn, and measured a suite of smoke taint marker compounds to see whether the Kalyn had any effect. What we found in this particular trial, what we also did was to monitor the volatile phenol concentrations after smoke exposure and also the volatile phenol glycoconjugates to try and get an understanding of how quickly the volatile phenols were being metabolized and how quickly these glycoconjugates were accumulating. And what we found was 1 day after smoke exposure, we were no longer able to detect these volatile phenols. And even though we were seeing some of the glycoconjugates 1 week after smoke exposure, we saw a significant increase in concentration from 1 week through to harvest, so 1 week through to 4 weeks post smoke exposure. So we weren't able to calculate a mass balance for what was going on with the volatile phenols we were measuring. And I'll come back to that in a moment because we followed up on that in a more recent study. But just to explain the Kalyn story, what we've got here is our Sauvignon Blanc data, and we can see low levels of the volatile phenol glycoconjugates in the control grapes. And then our smoke-affected grapes have high levels. That makes sense that they've accumulated the volatile phenols and then the glycosides. But then when we had a look at the wines or the grapes that corresponded to the Kalyn treatment, we saw no difference. So in Sauvignon Blanc, the Kalyn treatment had no effect. In our Chardonnay grapes, we saw a slight effect of Kalyn, but it was only in our Merlot grapes that we saw, sorry, that should be Cabernet Sauvignon. It was only in the red grape variety that we saw any effect of Kalyn at all. And when we went back and thought about what might have been going on here, the PhD student who did this work explained that in the case of the Merlot grapes, the coverage of the Kalyn on the grapes was really good. It stuck really nicely. But in the case of the 2 white grape varieties, it was quite patchy. And so we think this might have been related to coverage. So this is something that I've sort of earmarked as, we really need to go back and have a look at this in a little bit more detail. But at the same time, we're also mindful of the fact that when a bushfire occurs near a wine region, it's often a little bit difficult to just convince people that before they evacuate the area because there's a fire, they just need to quickly pop out to the vineyard and apply some Kalyn to the grapevines to prevent there being any smoke exposure and smoke taint of the vines. So it's something that might be worth having a look at later on, but whether this is really a practical solution for industry or not, we're not really sure. More recently, the trial that we have investigated was trying to see whether we could mitigate the uptake of smoke compounds in a vineyard during smoke exposure. So a number of preliminary trials have investigated whether we can just wash grapes following smoke exposure, and does that just wash the particulate matter that might accumulate on the grapes off and therefore, alleviate the issue of the taint. So one of my colleagues has developed this in-canopy misting system. So he has these sprinklers set up inside the grapevine canopy, and he uses it to mitigate the effect of heat waves. So, you know, when the temperature gets too hot, grapevines don't like to photosynthesize during heat waves, and so he can use this system to basically cool down the grapevine and ensure that the vines can continue to photosynthesize. So we looked at it and said, well, what if this washing will essentially drop out the smoke particulate matter during, you know, smoke exposure, and therefore prevent the uptake of smoke compounds during, you know, during a bushfire event. So we trialled this, and again collected samples at different time points to be able to have a look at the accumulation of glycoconjugates in the grapes over time. When we first got the grape glycoconjugate data at harvest, it looked quite promising. So our control grapes and our control with misting grapes had, again, low background levels of some of these marker compounds. Our smoke-affected grapes had different levels of different volatile phenol glycoconjugates. And it appeared to us that the grapevines that had been exposed to smoke whilst we were applying this misting had lower levels of these volatile phenol glycoconjugates than the corresponding smoke, which might have suggested reduced uptake of these compounds. Unfortunately, by the time we got through to a wine outcome, there was no statistically significant difference between the concentrations of volatile phenols in our smoke-affected wines and our wines that came from the smoke and misting treatment. And then when we looked at sensory analysis, the grey line is our control wine, so low levels of these smoky aromas and flavours, and the dark line and the dotted line are our smoked and our smoked plus misting wines, and we can see that they've both got very similar intensities of smoky cold ash aromas and flavours and that drying ashy aftertaste. So unfortunately it looks like the misting hasn't had any effect at all. This is still interesting because what it's telling us is that these volatile phenols are clearly taken up by the grapes incredibly quickly, such that, you know, dropping out any particulate matter or washing the grapes, even while we're applying smoke, isn't stopping them from these volatile compounds from getting into the grapes. But unfortunately, it's not the solution to smoke taint that we were hoping for. But the other part of this trial that was very interesting to us was monitoring the kinetics of what was going on with glycosylation. So whereas in the previous study where we'd done the Kalyn trial, we had measured grapes at 24 hours and seen that there were no volatile phenols, in this trial we included a time point where we sampled 1 hour after smoke exposure. So what we saw was prior to smoke exposure, really low background levels of these volatile phenols. 1 hour after smoke exposure, we can see, you know, elevated concentrations of glycol and syringol, and the same patterns were observed in the other volatile phenols that we measured. By 24 hours after smoke exposure, a significant proportion of those volatile phenols have been metabolised and are no longer detectable by GCMS. And we see that by the time we get to 1 week and to 4 weeks, those levels have dropped quite quickly. But what's really fascinating is that the accumulation of glycoconjugates doesn't follow that decline in volatile phenols. So whereas we might expect to see, and to some extent we have seen that with glycoconjugates in earlier studies, what we were seeing is some of these glycoconjugates were detectable even 24 hours after smoke exposure, but from 1 week through to 4 weeks, so time point 3 is 1 week after smoke exposure, time point 4 is harvest at 4 weeks after smoke exposure, there's a significant increase in these glycoconjugates. So that's interesting from a couple of different perspectives. Firstly, it leaves me thinking what's going on with our volatile phenols, what form are they in that we're no longer able to see them, you know, from 1 day through to 1 week. Does this mean that there are alternate storage forms? Do we have these volatile phenols being sequestered in other parts of the cells or in cell walls, or is there some other intermediate that's being formed as part of the glycosylation process? But it's also useful for industry to know because it means that at least in some grape varieties, there may be this window, possibly between 1 and 7 days post-smoke exposure, that if you go out and sample your grapes to send away for chemical analysis to determine whether you've got smoke taint, you're going to significantly underestimate what you will see either in the grapes at harvest or, more importantly, what you might see in the wine. So this is something that we're certainly investigating further, trying to understand both the biochemistry that's going on, and we have done some work looking at the activity of certain glucosal transferase enzymes in grapevines following smoke exposure, but also trying to look at alternate analytical methods to understand what's going on. So to finish off my presentation, I just want to talk about what the good news for industry might be. Firstly, I don't have any data to present to show it, but we know from multiple studies that there's no carryover of smoke taint between seasons. So even back with some of Kristen Kennison's really early work, where we applied smoke to those Merlot grapevines at 8 different time points, in the subsequent season, those grapevines didn't give us smoke tainted wine. This particular study, which I did with one of my viticulture colleagues, we went out into a vineyard to look at the recovery of vines that were scorched during a bushfire. So in this instance, not so much smoke damage to vines, but heat damage from the bushfire itself. And this has been a really useful study, given that some of the fires, particularly up in the Adelaide Hills, that have occurred this year. So what we were able to show is, and it depends on the level of heat damage, but certainly grapevines are capable of recovering following heat damage. And whilst the buds that may be damaged in one season may not produce shoots and therefore fruit in the subsequent season, the grapevine is able to respond, and 2 seasons after that fire occurred, we saw that yields and certainly vegetative growth as well was almost back to being comparable to vines that weren't scorched during the fire. So there is some recovery possible. The other good news, we've identified 2 methods that industry can use to at least partially remediate smoke tainted wine. So the first one makes use of reverse osmosis and solid phase adsorption technology. So essentially what the system does is takes a tainted wine and it fractionates that wine via reverse osmosis using a membrane that nominally has a separation around a molecular weight cutoff of 150 to 200 atomic mass units. So essentially small molecules like smoke taint, volatile phenols, will pass through the membrane, but larger molecules like our anthocyanins will be retained in what's known as the retentate. This allows us to selectively treat that permeate fraction so we can pass it over some kind of an adsorbent resin, which hopefully is going to take out our volatile phenols before we take the treated permeate and recombine it with the retentate. And so we investigated whether this system could be used with smoke tainted wine. And what we found was, as we progressively treated a smoke tainted wine, with time we were able to reduce the concentration of these volatile phenols. However, the volatile phenol glycoconjugates aren't being removed from this process, basically because their molecular weight is higher than the nominal cutoff of our membrane, so they don't permeate through the membrane and therefore aren't being adsorbed on that adsorbent resin. But in our trial, what we did find is a significant reduction. So we've got 2 examples of sensory data here. The dark line, or the solid line, is the smoke tainted wine, and the dotted line in each case is the treated wine. And so we can see that there is a significant reduction in the intensity of smoky cold ash aromas and flavours in those wines. So it's certainly one method that is being used by some wineries to treat smoke tainted wine. The other amelioration technique that we've had some success with is the use of activated carbon. So activated carbon just being an adsorbent in its own right, and typically one, as most chemists will know, that's not particularly selective. It's happy to bind lots of different things. But we had a particular activated carbon which showed quite good affinity for volatile phenols. And so we were able to take a smoke tainted wine, So this was the Merlot from our varietal trial. And we were able to significantly reduce the concentration of volatile phenols in that wine. And this is probably one of the first times where the blue line corresponds with our smoke tainted wine, and the red line corresponds with our treated wine. And it's probably one of the first times that I've seen such a significant reduction in the intensity of smoke attributes that we've also seen a corresponding increase in the intensity of fruit aroma and flavour characteristics. So most times we see a slight reduction in smoke characters and no effect on fruit aroma. Here it was such a noticeable difference. But again, one of the limitations, at least with the carbon that we used in this trial, was that it wasn't removing the volatile phenols, the volatile phenol glycoconjugates. And the Australian Wine Research Institute has done some work to show that they themselves might impart some sensory perception of smoke taint, because some of the enzymes in our mouths are actually capable of breaking those compounds down and releasing the volatile phenols. So some people will perceive that smoky character from those glycoconjugates. However, more recent work being done by the Australian Wine Research Institute suggests that there might be some activated carbons out there that actually are quite good at scavenging these glycoconjugates. So this is an area where more work is being done. So just to finish, and I haven't even presented all of the work that I've done on smoke taint today, but hopefully that's given you, a snapshot of what we have done. Some of the ongoing areas that we're still working in is methods for monitoring vineyard exposure in smoke. So for the past 2 seasons, we've been working with a company that has these commercial sensors. So they measure particulate matter, and we've been looking at how well those sensors can detect smoke exposure. So it's not so much about predicting the level of smoke taint in the grapes or wine that would come from a vineyard, but at least giving industry confidence about whether or not there was any smoke exposure, and then they can make a decision about whether they need to send samples off for chemical analysis. So at least if you know that there was no smoke exposure, or just a short period of smoke exposure at low density, then you might not have a problem. So even that in itself would be useful for industry in terms of decision making. Obviously, reliance on techniques such as GCMS and LCMS for analysis of lots of samples during a season is time consuming in terms of sample preparation and processing. So we're looking at the use of spectral techniques for rapid detection and quantification of smoke exposure in grapevines or grapes and wine. And then we're still working on other strategies for amelioration of smoke taint in both grapes and wine. So we've been trialling ozone treatment to see if that ozone can actually break down the volatile phenols or convert them into forms that have less sensory impact, and also looking at other additives that might be a bit more selective for either the volatile phenols or their glycoconjugates. So it would be remiss of me not to acknowledge the very large number of people that have been involved in this research over the years. So there have been a number of PhD students that I've worked with, plus some colleagues, both here at the University of Adelaide and the Australian Wine Research Institute, a number of industry partners, and obviously our funding bodies, what's now known as the Wine Australia, and also the Australian Research Council, and our ongoing research, which is being undertaken as part of an ARC training centre for innovative wine production. So I'm happy to take some questions, and thank you for listening.
Thank you, Kerry. Thanks so much for your fascinating webinar. It was really, I know, definitely I really enjoyed it, and I'm sure that is the feeling we have from what I would normally say from the floor. I would just like to, normally what we do at the end of questions is that we would present our speaker with a token of our appreciation. That's a little bit difficult at the moment, and typically we would present our speaker with a nice bottle of non-smoke tainted wine. But if you check your inbox, because I've just pressed send, you have a gift voucher which is going to appear there at the speed of, well, as fast as the electrons can carry. So thank you. The reason that I wanted to mention that first is because I have a class at 4 p.m. which is very, very soon. So I'm going to thank you again and open the floor to questions. It's just I may drop out if the discussion
No problem.
is lively. So, but thank you so much.
My pleasure.
If there are any questions, please take your microphone off mute. Remember, you can do that by pressing the space bar, and I'm sure that Kerry will entertain those questions for you.
I'd like to commend Kerry very heartily for what was a very thorough bit of work. I'd like to ask, I presume this has been recorded.
Looks like it, yes.
I'm sure many winemakers would be quite interested in seeing the recording of this. People like Bruce Terrell, who suffered quite a lot.
Can I ask a fairly silly question, I suppose, Kerry? But in terms of value, when wine is tainted, by how much is the value of that bottle reduced?
That's an excellent question. I've actually approached some of my economics colleagues to ask whether we can set up a project to look at exactly that. Some of the numbers that have been put out there would suggest that, so most people would be aware of the fires that occurred in Victoria, so the Black Saturday fires. Some of those fires have been estimated to have cost industry tens of millions of dollars in terms of the loss of fruit. But then it's not just the loss of fruit, you have to consider what that fruit would have been worth once you'd converted it through to wine, which obviously is a significant value add. So those Victorian fires have been estimated to have cost the Victorian wine industry around $100 million. This is where it gets a little bit difficult, and I want to be really careful about trying to put a dollar value on the impact for industry, because obviously we know that there have been some significant fires over the vintage that's just gone, but the numbers that I'm hearing is that it's only a few % of the total production across Australia that's been in any way affected by the fires. And I think we need to be careful how much we talk about this so that we don't essentially extend either the impact of the fires from heat damage or the smoke taint itself to essentially taint our entire wine industry, that we give the Australian wine industry a reputation for don't buy smoke. Certainly when you've got wineries that declare that they're not producing a vintage for a particular year, that's got to be a significant financial loss for them. So it obviously is important, but I think I'm not an economist, so I wouldn't want to put a price on it, except to say, I'm sure there are some winemakers that, have been heavily hit this year. The other thing we have to recognise is this is by no means an Australian problem only. So wine regions in California, Oregon State, Washington State, in British Columbia and Canada, there were some fires a few years back in New Zealand. There have been some fires in Europe, and also the Stellenbosch wine region in South Africa. So this is an issue that's affecting wine regions around the world. So yeah, I think we just need to be careful how much we attribute this to being a problem for the Australian wine industry. And obviously it doesn't happen every year either. So.
Thank you.
Kerry, hi, I'm Elise. I just had a question for you around the experiment. I wanted to ask you about the types of fuel, as in what woods and things you use to do the smoke preparation, and also whether you looked at things like radiant heat and versus the effect of that and the smoke, or just one or the other on the grapes. and how much of the fuel load in the region, the types of fuels such as eucalypt and other species made, if there was any difference?
So those are all excellent questions. Pretty well all of the work that I've done and all of the field trials, right from the early days with Kristen Kennison through to the trials that we're still doing now, we're using barley straw as a fuel simply because it's relatively easy for us to ensure that its composition is the same and that we're burning similar quantities and producing smoke in the same way. So we do get some interesting questions when we go to publish our work about how reminiscent is the smoke from barley straw relative to, you know, other fuels. And it's a good question. There has been some further work that was done at Curtin University that compared the composition of smoke from different fuels. And irrespective of what they were burning, they were still seeing that these volatile phenols were the predominant components of smoke. Some of the other work that's being done elsewhere in the world, so there's some work being done by the University of British Columbia where they've produced a fuel that's a bit more reminiscent of what might burn. So they're collecting leaves and twig material and then making essentially like a brick that they're burning. But again, I'd argue that's still not reminiscent of what's happening in an actual fire. So it's kind of a trade-off for us in terms of being able to conduct controlled and replicated experiments versus burning something that is maybe more representative of the fuel that burns in a fire. So in the last year or two, we have been doing some work in this area, looking at smoke from different fuels, and, you know, consistently with some of the other projects that have been done, we are seeing that it's still a lot of volatile phenols, irrespective of what we burn, that are being produced. And what's really interesting is some of the early work that was done looking at the influence of smoke on seed germination. They moved away from burning different plant materials and were burning Whatman filter papers because that was still generating the volatiles that are responsible for seed germination. Yeah, I'm not sure I've covered off all of your questions in that, so let me know if I've missed something.
I think it's just the other one was around radiant heat and whether like a more intense heat made any difference.
So we have tracked the temperature in our smoke tents during those experiments, and we know that in the course of a 1 hour exposure, and obviously it depends a little bit on the ambient temperature on a given day, but we typically see less than a 10-degree increase in temperature inside the tent. We have done some work with plant physiologists who use thermographic cameras to have a look at changes in heat in the cameras, in the grapevine, and they do see some increase in temperature, basically because the vines close the stomata of the leaves and stop some of that photosynthetic activity, so they're not cooling as part of that process. We have done one experiment where we did apply heat as well as smoke, but that's the one trial that my postdoc from that time hasn't written up, and I haven't seen that data yet. So it's the one study. My gut feeling is by increasing the temperature at least a little bit, you might be promoting some of those biochemical pathways that allow the uptake and metabolism in the berry to increase. So it might give you greater higher intensity of smoke taint, but at some point, you know, the vine, if it gets too hot, it's going to stop photosynthesizing. So some of those biochemical pathways will actually shut down. I think even the uptake of smoke itself, so how these compounds are getting into the berry, is still a little bit unknown. So people often say, oh, isn't it just taken up by the leaves and then translocated to the berries? Whereas I think the isotope tracer experiment that AWRI did suggests that there's probably not much, if any, translocation going on. So my gut feel is that it's just these things are directly absorbing into the berries. But one of my colleagues has said, have you thought about the uptake of these compounds through lenticels in the essentially the stem of the berries, and I had to quickly go and look up what's a lenticel, and it's essentially like a stomata of a leaf. It's a pore that allows gas exchange. So that's sort of the next step that I want to have a look at to see whether that might be a pathway for these compounds. But my feeling is that it's still just direct absorption across the surface of the berry itself, which might explain some of the differences we see in varieties, that berry skin thickness or the waxy layer and how that changes during the growing season as the fruit's ripening might be what's influencing the uptake.
Thank you. Yep, very comprehensive.
Well, I think Robert's actually had to leave us. So I guess, can I ask, are there any more questions? Looks like the answer's no, in which case I will, on behalf of everyone here, say thank you again to Kerry for a really fascinating and enlightening presentation. It's something that I think we all have a great interest in. I mean, I guess most of us probably drink wine, but it's interesting to know the work that's been done to determine the impact of smoke on grapes and wine. Thank you again, Kerry, for joining us and for making your presentation this afternoon. Thank you for everyone here for participating. Thank you especially to Elise from the RACI for ensuring that everything works smoothly. And I guess just say goodbye. And the Food, Nutrition and Analytical Chemistry group of the Victorian branch of the RACI, that's a bit of a mouthful, is planning some further webinars or meetings, virtual meetings, later on in the year. So keep your eye open for news about the next seminars and presentations that we have. So I think with that, we'll say goodbye. Okay.
Thank you.
Thank you. Bye.
Bye. Bye.